recorded. And let's just make sure this works okay. Ask good questions. All right, so the OH, the one we were talking about that is making the difference, is that one right there and that one right there. That's the difference between the two. And it could be pointed up or down. Those cyclic forms are called Haworth forms. It's called a Haworth projection. And we're going to learn in just a few minutes how to draw it. It's kind of mysterious how you go from the straight chain to the cyclic and then have all those groups going up and down correctly. It's just a little bit weird. It actually is not hard to do at all if you follow some set rules that I'll show you and never deviate from them, and then you'll be okay. Now what's going on to close it up is this last OH, this last chiral OH, is actually coming around and attacking the carbonyl. And so you're forming, an OH attacking an aldehyde was forming a hemiacetal. Remember that reaction? We had OHs, alcohols, attacking ketones or aldehydes, and it was forming hemiacetals. And that's exactly what you're doing here. So the product of that closure is an OH, and when it closes, it could be up or down, depending on if the attack was from the top or from the bottom. Okay? That makes sense to everybody? So that is why that is the only one that's varying. It's the only one where it could be one or the other. All the other groups are kind of set. Okay, so these Haworth forms, they are related, of course, they're in equilibrium with each other. They are diastereomers. I'll just abbreviate it. And since it's sugar chemistry, they have a special name, because everything in sugar chemistry does. Instead of just calling them diastereomeric Haworth forms, they call them anomers. Anomers. And it is spelled A-N-O-M-E-R-S. They are called anomers. And everyone knows if I say, oh, it's the anomer of this, what I'm talking about is I'm only talking about the carbon where that OH could have been up or down, the carbon where the reaction occurred for closure. Okay? So that carbon, carbon number 1, which is the aldehyde carbon, carbon number 1 is the aldehyde carbon. Since it gave us the anomers, it's called the anomeric carbon. Okay. Now we have to have a way of designating which one is which. Something easier than kind of waving your hands and say it's pointed down, it's pointed up. That's not going to work. And so what they do, if the OH on the anomeric carbon is cis to the CH2OH, like this one right here. There's the OH, there's the CH2OH, they are cis. That is called the beta anomer. If the OH on the anomeric carbon is trans, so that's the second one. There's the OH, and it's down, the CH2OH is up, they're trans, then that is called the alpha anomer. And I had the hardest time, I still do, remembering this. I do not know why. I cannot remember this to save my life, but there's a trick. Okay, here's the trick. See if it works for you. Beta, if you're drawing a beta carefully, watch me draw it. We go like this, beta, and it ends up. So they're both up, right? They're both the same way. The CH2OH, the CH2OH is up in the other one, and so watch when I draw an alpha. The OH is pointed down. The alpha ends in a down. And I have to, I swear to you, every time I go think about this, I have to think about that. So if it's beta, the CH2OH, we're going to, in a D sugar. In a D sugar, CH2OH is always up. So, and D sugars are the natural ones. We're going to see the most of them. So CH2OH is always up. That means if something's beta, the OH is also going to be up. Beta ends in an up swoop. CH2OH is always up in a D sugar. If the OH is down, its alpha ends in a down swoop. Okay, good luck with that. We'll see how that goes. Okay. So far, so good. Any questions? All right. Next slide. We're going to deal with fructose now. Fructose is a little bit different because it is a ketose. So there's a ketone group, and so it's going to look a little bit different. When it closes up, we'll go to our last OH, and it is going to come loop around and close in on the ketone. And if you count the number of atoms in the ring, you're going to get a 5-membered ring. So 1, 2, 3, 4, you end up closing up in a 5-membered ring. And so you can see the two Hayworth projections there. The CH2OH in both of them is up. Okay, so that's a D sugar, so the CH2OH is up. And the trick with this is finding the anomeric carbon. That is a little bit hard to do. If you take a look, the anomeric carbon came from this carbonyl right there. Came from that. That carbon will have an OH on it. Once it gets attacked, it'll have an OH. It'll also have a CH2OH on it. So you've got to find the carbon in this ring that has an OH and a CH2OH on it. Do you see it? See that one right there? You guys okay with that? And that one right there. So the oxygen that attacked, the attacking oxygen is, I'll put a box around it right there. That's the attacking oxygen, that's the attacking oxygen. It attacked the former carbonyl carbon, which is that right there with the arrow pointed to it. That's going to have an OH from the carbonyl, and then that CH2OH is just hanging out because it's attached. And then we see that if the OH is cis to the CH2OH, beta swoops up, right? It's pointed up. And so that's the beta form. And then when the OH is down, that's the alpha form. So ketones are a little bit more strange because the anomeric carbon has stuff on it as well. So it's a little harder to identify. Yes? The arrow is pointing to the anomeric carbon. Everyone okay with that? Yes? Okay. All right. I can't tell if you are or not.
Could you go, why is there ether in there again?
The ether being this part right here?
Yeah.
Okay, what that is, that oxygen right there, is this last chiral oxygen, this last OH on the last chiral carbon. It comes around, it attacks, electrons come up, that makes this an OH. Good with that? Okay? When you do minus H plus plus H plus,
Yes.
this oxygen loses that H. Okay? So then it is in the ring. It's part of the ring. You're looking at that oxygen right there. This oxygen is this oxygen right there.
Okay.
Okay? Because it went looped around. Yeah?
So when you look at the bottom from CH2OH, that one is alpha?
This bottom CH2OH right here, which came from this group right here, up here, you know, I don't know that they don't refer to it. They refer to the anomeric OH and its relationship to that last CH2OH here when they're doing the designation. Okay, otherwise if you tried to designate both, you wouldn't know whether to call it alpha or beta. You have to focus on something, and they focus on the OH. The anomeric OH. Okay. Now the fun. How to convert a Fisher to a Hayworth. There are all kinds of ways to do this. And, you know, I'm a spatial genius, and so I can't do most of them. I know the book I learned it in, they talked about, like laying it on its, what'd they do? They just said you just loop it, and then if you like twist it and rotate it, you can obviously see that this is this. I couldn't do it. So I have a set of rules. This is how I do it. It's the only way I can do it. And it's foolproof because I can do it. So what we're going to do, you guys just do it exactly like this. It doesn't require any spatial geniusness to make it happen. And so what we're going to do, going through this, we're going to convert our D-glucose into a Hayworth projection. So this is, right here where it's labeled, this is normal D-glucose. Plain old normal. The first thing you do in this process is you rotate the groups on the last chiral carbon, so that's that chiral carbon right there, I'm going to rotate them so the OH is at the bottom. So what that means is I took and I swung the OH down, the CH2OH rotated up, and the H rotated over. That's like taking the molecule, if you imagine a model kit, grabbing that last group of atoms, that last tetrahedrocarbon, and just rotating like this. Okay, remember you have to rotate everything. I cannot just take the OH and swap it with the CH2OH. Because there's no way in a model kit to take a carbon with 3 groups and change 2 of them and leave 1 alone without actually breaking bonds, right? If I'm rotating, they're all moving, right? I have to move everything. So we're going to move the OH down, CH2OH goes up, H goes over. That gives us our second picture. So now the OH is down. Then what you're going to do is you're going to drop the whole molecule on its side, drop to the right. So this thing crashed like a tree to the right. So far so good. Okay, not too hard. And then you close it up. So the OH comes along, hits the aldehyde, and that gives me my anomeric OH and the oxygen in the center. All right, next slide. Existing, whatever. Okay, so what I've got is I've got my 6-membered basic ring. When I close this thing up, the oxygen, by convention, it always goes in the upper right-hand corner, like that. Okay, so that's the convention. So I've got that. The anomeric carbon, which is this carbon right here, where the oxygen is now attached to, that is going to bear an OH. That OH could be up, or it could be down. And so I haven't told you which one it is. So what we're going to do, that's the purpose of the squiggly line. When you see a squiggly line, that means either one. Okay? And if I were to tell you alpha or beta, you could then be a lot more specific, but we aren't saying right now. So it's just a squiggly line. And now, what you do, when you're placing the other groups, the groups that are up, right there, they go up on your ring. The groups that are down, right there, those go down on your ring. So you just go around this ring and start putting up, down, as it shows on your sideways molecule. So let's go ahead and let's do it. The first OH, as I go around, so that's that one right there, where I just put the dot, that is down. So OH goes down. The next OH down the chain, up. OH goes up. The next OH on the chain, down. OH goes down. CH2OH, up. And there's the molecule. Now if I were to tell you alpha or beta, you could then take that anomeric OH and put it in the right spot. Okay? Yep. Now, we're going to have to be naming these cyclic sugars. I think that might be one of the ACS sort of questions, is having you look at one and tell something about its name. To name a cyclic sugar, it's really pretty straightforward. The first thing you have to specify is the anomeric carbon, how that OH is. So you're going to specify alpha or beta. That's an important piece of information. Then you have to specify, is it a D sugar or an L sugar? And then the name of the sugar, like glucose, maltose, whatever it happens to be, you have to have its name. And the sugar name, the sheet I'm going to give you, they all name an ose, dextrose, maltose, whatever, glucose. What we're going to do is we are going to drop the SE ending, and that'll leave you with, say it's glucose, leaves you with gluco. We got rid of this part. And we're going to then tack on the ring type. Is it a 5-membered ring or is it a 6-membered ring? Most will be 6. Most will be 6. So, this particular example that we have right here, we're going to name it, but we have a fundamental problem, and that is that we do not know what sugar it is. Can I tell you if it's, or can you tell me, if is it alpha or beta? You guys are good. Yeah, it's alpha, okay. Is it a D or an L sugar? The CH2OH is up, so D sugar. Okay. Parent sugar name? Beats me. I don't know. So we're going to have to, guess what? Peel the thing apart. Okay? So to do that, let's go ahead, and I'm just going to go to the board for this. So to take it apart, let's draw it in its sideways form. Aldehyde is here. OH is at the end. 1, 2, 3. Do I have enough carbons? 1, 2, 3, 4, 5. I will have enough carbons. Okay. We're going to put the groups up and down as they should be. So my first OH going down the chain from the anomeric carbon is down, right? So it's right there. And then up, and then down. And then CH2OH is up, H was down. Everyone okay how I'm doing that? I'm just dissecting it, right? Just dissecting the ring. Okay so far? Yeah.
How could you tell if it's D or L from just looking at that?
Oh, because when you do a D sugar, when you do that rotation around the bottom, the CH2OH is always going to end up on the left. And then when you drop it sideways, that's always the up position. Okay, let's stand it up right now. And we have OH, 1, 2, 3, 4. So I'm tipping it counterclockwise, essentially. So it's going to be OH there, OH there, OH here, and CH2OH here, and H there. So far, so good? I still can't name the sugar, though, because in order to match it to what's on your sheet, CH2OH has to be on the bottom. So now we're going to rotate out of it. We're doing everything backwards that we just did. So CH2OH will rotate to the bottom, OH over there, H there, and I have 1, 2, 3, 4 chiral carbons, OH, 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 and the OH ended up there, the H ended up there. That is the sugar that it started from. So look on your sheet and find out who that is. It's along the bottom somewhere.
It's Maryland?
I found it. You guys see that it's glucose? Okay. So this is D-glucose. So if you're given the cyclic sugar and they say to name it, you have to rotate. Basically, peel everything apart, rotate again, and figure out what sugar it is. So now we're ready, finally. You told me it was alpha. So we have alpha, D sugar, D, gluco, and pyranose or furanose? Pyranose. There we go. It's an alpha D glucopyranose. Okay, that's not too bad. Okay, now see what you can do with this. We're going to draw the Hayworth projection for this sugar, the beta anomer this time. Then we're going to put it in a chair conformation, because sometimes you'll see them in chairs. And then we're going to name the cyclic sugar. And do a normal 6-membered ring sugar, okay? The only time we'll really see 5s is when there's a ketose. All right. So you guys spend a minute doing it. And then we'll quickly go over the answer together. stealth mode and put the answer up. Okay, let's see if I can get the image back so we can check our work. It came back there. I was very sneaky, wasn't I? Okay, so what I did here is I rotated around the bottom to get the OH at the bottom, and then dropped the thing on its side. And when I did that, the top two OHs were up, and then down, and then the CH2OH was down. It's a little bit different. And then we are going to close the sugar up by having that last OH attack the carbonyl, right there. We get our cycle. By convention again, the oxygen, the attacking oxygen will go in the upper right. Yeah, we'll talk. We'll see if I guess right. Ignore the anomeric carbon for just a minute. We'll check that out. The CH2OH was down. Everyone okay with that? Okay. The next OH was down. Then up and up. And then it said beta. Now beta by definition was having the last chiral OH, or the OH on the anomeric carbon, cis or trans to the CH2OH? Cis. So I have to draw it cis. The CH2OH is down this way. It happens to be down. It has to be cis. This one was a little weird. What made it weird? It's an L sugar. Exactly. If you take a look at the sugar, the OH on the last chiral carbon is on the left. So it's an L sugar. So things were a little weird. Okay, now redraw it in a chair. Don't get intimidated about the chair. It's actually pretty straightforward. What you'll want to do is go ahead and just draw a chair, just a skeleton of a chair. There it is. In the upper right-hand corner of your chair, put an oxygen. So there's my oxygen. And now you just go around and put the groups up or down, up or down. They may fall axial or equatorial, just depending on what that position dictates. So, the anomeric carbon, which is this bottom part of the chair, like the foot part, the OH is down. That happens to be axial. So there it is. Then the next one, the OH is up. That happened to be axial as well. Then the next one, going around, the OH is up. Equatorial. And then the CH2OH was, I've lost, let's see, hang on. There's the anomeric OH, there's the anomeric OH, then we go up, then we go up, here we go, the OH, we're at the OH now, it's down. There we go. I had skipped that one OH. Then we get to the CH2OH, it's down. We've got to squeeze it in there. I'll squeeze it in like that. There it is. There you go. So the chair form's super easy. You go to a Hayward first, and then just draw your chair, put the O in the upper right-hand corner, and up, down, up, down, up, down. As needed. Okay? You're likely to see them in all these forms. You'll see straight chain, particularly when we want to do reactions, because the reactions usually center around the aldehyde. So you'll see straight chain. You'll see the Hayworth sometimes, and then sometimes you'll see a chair, and you just have to be able to kind of manipulate all three. Yeah?
Can we move the mass range into the attacking oxygen down?
The one, let me just circle it to make sure. This, are we talking about this one right here? Because I told you it was a beta sugar. I said draw the beta anomer. Okay, the anomeric carbon you don't know. It could be either up or down. The beta anomer is where the OH is cis to the CH2OH. So I knew it had to be down since CH2OH was down. Okay, otherwise you wouldn't know. If I hadn't told you alpha or beta, you wouldn't know. All right, name this thing. All right. Well, we know it's a beta, right? So our name, we have beta. We know it's an L sugar. Do we know what sugar this thing is? That's sort of fun, huh? Because on your sheet, what kind of sugars do you have? D sugars. So this problem just goes on and on and on, doesn't it? What are we going to have to do? It's an L sugar. We got to find out what its enantiomer is, right? That's the D sugar. So take the sugar, draw its mirror image. CHO. Here's CH2OH at the bottom. Every chiral carbon, we're going to draw the mirror image of this thing. So if the OH, we're going to work up the chain. Let's work up the chain. That last chiral OH is to the left there. It's to the right here, right? The one that was to the right is going to be to the left. The left goes to the right. And the left goes to the right. Is that the mirror image? And this is a D sugar. What sugar is that? I have no idea what sugar it is. I don't have these memorized at all. Oh, is it glucose? That would be boring. No, it's not glucose. Is it, let's see, to the right, to the right, to the left, to the right. It's gulose. G-U-L-O-S-E. Yeah, it's the one that does D, G-U-L-O-S-E. So that means the one that is sitting there on your paper is L-gulose, right? It is the mirror image of D-gulose. So that's L-gulose. So now we're getting close to our name. We know it's a beta sugar. We know it's L. We know it is gulo. We're dropping the part. So gulopyranose. There's its name, finally. Right there. These aren't hard. They do take a little while to do. Okay? Doing good? Excellent. Okay. All right. Now, we talked about how you can have a straight chain form or a cyclic form. And the cyclic form is actually about 99. The two taken together, those cyclic forms, are roughly 99% of that equilibrium. So they are definitely the predominant thing. 1% is about the straight chain form. We're going to see in a few minutes the reactions actually occur in that straight chain form. So it's important for a sugar to do reactions that it be able to open up. That's kind of critical. If 99% of it is sitting there looking like that, but I'm trying, say, to oxidize the aldehyde, in the cyclic form there is no aldehyde. I can't oxidize an aldehyde in that. It has to open up, and then I can oxidize. Okay, so we're really going to be relying on that thing being able to open up and give us the straight chain form. The equilibrium between the straight chain and the cyclic forms is an equilibrium between straight chain and alpha and straight chain and beta. And that process, that interconversion between the alpha and the beta, going back and forth, is called mutarotation. Mutarotation. So we can talk about sugars mutarotating, and that's what we're talking about, is going alpha to beta, beta to alpha, back and forth. It's a mutarotating. You're not going to get 50-50 between the alpha and the beta. It's going to depend on like how many groups are axial in each form, because remember, axial is not as good. So if beta had less groups axial, then that might be the preferred form. And it could also depend on like steric effects, things like that. So it's not going to be 50-50 necessarily. All right. Moving on, we're into reactions. There are, of course, a number of reactions that you can do. A lot of them are driven not so much by getting your sugar synthetically to react so that you can make something else with it. That's typically not what they're doing. Usually, when you're doing these reactions, to try to figure out what kind of sugar you have, to try to get an identity. So if I have some unknown sugar and I want to know what it is, I start doing a bunch of little reactions, typically in a test tube, to figure out, okay, is there an aldehyde or is there a ketone? And is it this or is it that? That sort of thing. And so it's a lot of it that's identity driven. So the first one is oxidation. And in oxidation, we are going to be taking the aldehyde and converting it into a carboxylic acid. Now you have to be a little careful. You guys can absolutely do that. We have all sorts of reagents that can do that, right? The problem with your reagents, though, is that you have, what, 5 alcohols sitting there? So if you use your standard chemistry, you're going to oxidize the aldehyde, and you're going to oxidize those 5 other alcohols, and it's going to be a carbonyl party. And you don't want to do that. And so they do not use the common reagents that you use. They use these really weird, wimpy ones. And so the list, I gave you guys a list of 4 of them. These are fairly weak. They're not going to go wild and oxidize everything. They will oxidize only the aldehyde, only that CHO group. Okay, so that's why they're kind of weird. I am not going to have you memorize their names. Okay, bromine water, I expect you would know. But the others, Benedicts, Phthalines, Tollens, the important part to see is that they're all metals. We're interested in this metal part right here. And these oxidations are actually something that are done where you put the sugar in a test tube, you add a little bit, say, of, I don't know, Benedict's reagent, shake it up, and you watch it. You just look, that you get results that you can see if there is an aldehyde. So let's say there's an aldehyde, and I use, I don't know, let's use Tollens right there. So I have the sugar, and I want to know, okay, is it an aldose or a ketose? Well, I put the sugar in a test tube. I add some Tollens reagent. If the aldehyde gets oxidized, then the reagent gets reduced. Does that make sense? So this would get reduced. If it gets, I squeeze a C in there. If it gets reduced, the silver plus gains an electron, and it becomes silver with no charge, silver metal. And so what you'll see in your test tube is if there's an aldehyde, if it gets oxidized, therefore reduces your reagent, you see a silver mirror build up on your test tube. Your test tube actually gets a coating of silver on it. For that reason, they call Tollens the silver mirror test. If you have copper in there, what will happen is those copper reagents will get reduced, and then you start seeing copper precipitates. You see kind of a reddish precipitate if it gets reduced. So you look for things. You're looking for silver to precipitate. You're looking for copper to precipitate. These brick red kind of precipitates. If you get those, you know you have an aldehyde. If you don't, you know you didn't. Okay? As far as we have a blank there, let's put some names in. You may be asked this. If it's the aldehyde form, it's called an aldose. If it is the carboxylic acid form, if it gets oxidized, this is just bizarre. They call it an aldonic acid. It's a carboxylic acid that came from an aldehyde, an aldonic acid. There you go. Okay? So in order for this to happen, you have to have that aldehyde present. If it is a ketose, and there's a ketone, this won't work. Okay? There is one more thing, though, that could mess this up. We talked about how the sugar is mostly cyclic, right? If it for some reason gets locked in that cyclic form and is unable to open up, there's no aldehyde in the cyclic form either, so this would be negative. Okay? That's what the next slide's talking about. So, to be oxidized, the cyclic sugar must be able to open up. Okay? So when you say it must be a hemiacetal, what that is saying is that it can open. So I have to have, and let's just circle the grouping that's the key grouping right here. I have to have that grouping right there. If I have that, then I can reform the carbonyl, and this guy can pop open, and I'm back to my straight chain form. If, for some reason, this OH, the hemiacetal OH, is blocked up, something is bonded to it, there's something there. This can't open up anymore. It has to be a free OH. So the key thing is this anomeric carbon must have a free OH. Highlight that like 5 times. You have to have a free OH. So this example down lower is an example of a sugar that would fail the test. Because if you take a look, some reaction has gone further. It cyclized, and then we did something to cause that OH to have a methyl group on it. Right there. Now it's not an OH anymore, and it can't become an aldehyde like this. This is now an acetal, and it cannot open. There's no straight chain, no oxidation. Okay? Now, here's the last strange vocabulary word for the slide. Sugar chemistry is full of strange things. If a sugar is oxidized, it had the aldehyde, right? If it's oxidized, it reduced something else. Okay? So sugars that can't be oxidized are called reducing sugars. I do not know why they couldn't just keep it simple like we've done our whole 2 semesters. But reducing sugar. If a sugar is a reducing sugar, it was able to be oxidized. It had the aldehyde. All right? For some reason, I don't know why it is that really annoys me, but that they do that. But any questions so far? Yeah.
Can you say that anomeric carbon should be neutral rotate as well?
Anomeric carbon what now?
Should be neutral rotate as well.
Yes, it should be able to mutarotate. If we had, like this one here where I have circled it in red, where it's OCH3, that couldn't mutarotate. It'd be locked into its alpha or beta form there. Is this the alpha or beta form? It's the alpha, and it's locked. It can't mutarotate, because to mutarotate, you have to go through the straight chain, and it can't. It can't get back to it. There's no mutarotation there either. All right. Now, this next slide is going to show us how to, if we wanted to, lock up that anomeric carbon. So what we're doing here is we're forming this thing called a glycoside. A glycoside is where you take and you essentially lock the anomeric carbon. We're going to lock that anomeric OH. And we are going to make it so that thing cannot open up. A glycoside is a word for an acetal from a carbohydrate. So it really is an acetal. And let's do a quick review on the board, because I don't know that with a sugar it's so easy to see how that is an acetal when we do it. So let's go ahead and just do a really quick drawing, skeleton drawing, of a hemiacetal and an acetal. If I had a carbon and, I'll just do like that, it's a carbonyl. If I had one alcohol to it, then it becomes an OH, and I have OR. Right? I've added just one alcohol to it. Okay? This is a hemiacetal. If I add two alcohols to it, then I get rid of the OH. It would leave as water. Remember, I protonate it, we do all that, it leaves as water. And I've added two alcohols to it, and this is called an acetal. Okay, we learned that, I think, back in Unit 2. Now, as it applies to a sugar, what we have with a sugar is we have a structure, when it first closes up, here's your skeletal structure, here's your anomeric OH, and let's just draw it like that. That is a hemiacetal. To see that it is, just take a look at the straight chain sugar. There's the aldehyde, there's all your groups down here. Here's your last chiral carbon. And what we're doing is we're taking an alcohol, this last OH on the chiral carbon, attacking the carbonyl, electrons go up. We're adding one alcohol to a carbonyl. And what you've got, this carbon right here, is a hemiacetal. It's got an OH on it and an OR. This whole chain is your R group. OH, OR, so it's a hemiacetal. If I want to turn that into an acetal, what I'm going to have to do, here's one OR group, right? I'm going to have to take that anomeric carbon and make that OH another OR. Then that is an acetal. Okay, so we all oriented all right? Okay, so what we're going to do in this next slide is we are just going to take this molecule and we're going to treat it with an alcohol. We get to choose which one, and an H+ catalyst. You can do methanol, ethanol. This particular case, they chose ethanol. And what that does is it takes that anomeric OH, which was the aldehyde OH, and it turns it into, and it's the only one that does this, turns it into an acetal. The others don't do it because they would essentially be forming, I guess, ethers. They aren't acetals. And so we can do them, but we can't do them that way. Okay? That's a reagent to make an acetal from a hemiacetal. And there's just one hemiacetal in that molecule, and that is at the anomeric carbon. Okay, so we have these acetals. They are just like a normal acetal that we're used to from like acetone or something. They are, first off, stable in water. They are not in equilibrium with the open chain form. That is kind of important. So what that means is that they cannot mutarotate. If they can't open up, they can't close the other way. They are stuck just like they are. So they can't mutarotate, and they can't be oxidized. They are non-reducing sugars. I need that aldehyde to oxidize it. I can't get it. So it's a non-reducing sugar. If I want to convert it back to a normal hemiacetal, H+, H3O+ is all that would be needed. Okay? And just hydrolyze it. Now to name the thing, it's going to name just like a cyclic sugar, but we need to specify the alkyl group that is on, that we just added to make the acetal. So in this case it'd be ethyl. Okay, you specify that group. Then you name the cyclic sugar like usual. you replace ose with oside to show that it's a glycoside. So, this guy right here, I'm going to give you its sugar name. The cyclic sugar is mannose. This is D-mannose. Okay, so if that's D-mannose, then the acetal, or the glycoside that I made, okay, here we go. We got to put ethyl in front because we stuck an ethyl group on there. Is it alpha or beta? Up, it's beta. Beta, D or L sugar? Well, I just told you it's a D sugar, huh? And it's going, so it's D, yeah. And it's going to be mannose. Whoops. M-A, let's go ahead and erase that. There we go. M-A-N, come on. M-A-N-N-O, mano, because we got to drop the ose part. So mannopyranoside. So it came from beta D mannopyranose. Now it's a aldehyde, so it's a pyranoside. Mm-hmm.
So when you treat this for H plus and ethyl,
Mhm.
why is it like the other sugars? Why is that specific?
Oh, because when you go through the mechanism, the question was why didn't it do the other OHs? When you actually go through the mechanism of this thing occurring, we had for our hemiacetal, it's just an OH, right? And we protonate with H plus, and it forms OH2, right, plus. That has to leave, right? And what the impetus for it leaving is the fact that this can come down and kick it out, right? That lone pair on the OR group that I've already added. None of the others have that. If the alcohol left on the, or the OH left on the others, it just leaves an unstable carbocation. So it doesn't do those. It only is going to do the hemiacetal part. All right, continuing on. Formation of ethers. This is doing all the other ones. And the reaction's actually pretty straightforward to do them. We're going to take and we're going to form OR groups at all those other OHs, every single one of them. So the first thing you do is you treat it with base, and the base will come and it will attack the hydrogen of that OH group, and it deprotonates it. And then you treat it with an alkylating agent. This is a methylating agent. It's dimethyl sulfate. And so the O- attacks dimethyl sulfate in an SN2 fashion. It's an SN2 reaction. Kicks out a leaving group. And I have added a CH3 to that oxygen. And then you do it again and again and again. And once you are completely done, you end up with this bottom one, where every single one, let's do this one here first, talk about that. Every single OH that was normal, that was not an acetal, has an OCH3 on it. Every single one. So those are all equivalent. Those OHs that I have in black are all ethers. There's nothing real special about them. They are ethers. The one, I'll do it in red. This one right here, the one in red, is an acetal. It's different. And what makes it different is this part right here. This carbon has two OR groups on it. None of the others have that. This carbon right here has an OR from the ring and the OR right here. These others are just R-O-R, just a plain boring ether. So if you expose this to acid, acid does nothing to ethers. They're stable. They sit there. But acid will hydrolyze the acetal. So this H3O plus step is where we hydrolyze the acetal. The only thing that's affected is the anomeric carbon right there. Now you might ask, why would you do this? There's a reason. Say you have, and I don't know if it's on the next slide or not. Let's take a look at the board. Yep, we'll jump to the board real fast. Okay, say you have a disaccharide. So I have two sugars, and they are bound together somewhere. Don't know where. Okay? So I'm going to draw one sugar. Just make one up real quick. And here's its O, and I don't know. It has OHs here, there, and everywhere. And it is going to be bound to the other sugar through, I don't know. Let's just draw the other sugar real quick. I don't know, through that oxygen. So their link is right there. Okay? And this one has an OH there, OH there, pop in all these OHs, and I will put this one up. Okay? And let's make this other one just the same. OH, and this is up, and then that's down. So this is my disaccharide. And I pull it out of a plant or something, I don't know where I got it, but there it is. And I want to know where it's linked, because I don't know. To figure out where it's linked, I need to know what's going on right there. Notice the linkage. The linkage is the anomeric carbon here to kind of a random OH there. You guys see where that is? The anomeric carbon of one is bound somewhere on the ring to the other. Okay? So what you do to figure out where it is you methylate everything. So I'm going to turn this into an OCH3, kind of protect it. Okay, that makes it an acetal. Then I take these others, and I take and put methyls everywhere. So there's a C, there's a C, there's a C, a C, a C, a C, and a C. Everybody's methylated. Okay? Then what I do is I hydrolyze this thing. I treat it with acid. And I am going to end up, cleave right there, what I'm going to see is in this sugar right here. So I have to do, there's some separation steps, of course. But what this is going to have is it's going to have that acetal turns back into a hemi. The ethers are unaffected. So that's an OCH3. That's an OCH3. This one here, OCH3. All these are unaffected. Nothing's happened. That one right there, what is it going to be? It's going to be an OH, right? Does everyone see that? It never, ever got methylated. So this one right there is an OH. So you separate these sugars, pop it into an NMR. You know, you have correlations. You know which peak belongs to which CH3 or whichever. I look at this and I say, look at that. At carbon number 1, 2, carbon number 3 didn't get methylated. I know my link has to be carbon 3 then, because it wasn't available, which means it had to be tied up. Make sense? So you methylate everywhere, then you cleave it. The one that didn't get methylated had to be where they were hooked up. Okay? That's what that's used for. Yeah. The other one would look like, the other half would have an OH there, because that would come off, and then all the rest of these are ethers. So they're just unchanged. So you'd have to do some separation. But it was like a column. You could do it in a column, because this has 2 OHs, that only has 1. They'd separate. Okay, so the ethers never get affected. And by seeing which one didn't become an ether, you know it was tied up. Okay, so that's useful for figuring out where linkages are, is where that's done. It doesn't really help you so much with sugar identity, though. I couldn't tell you what the names were of the two things linked, but I could tell you where they were linked. This next reaction here, reduction, helps you out again with trying to figure out what individual sugars are in this thing. So reduction, again, we're focused on the aldehydes, the only thing in there that can be reduced. and the aldehyde will be turning into, just as you'd expect, an alcohol. Okay, so the aldehyde gets reduced, turns into an alcohol. Any of the reagents we use are fair game because we can't hurt the things that are already alcohols, right? It's not like we're going to obliterate them. Okay? So, the advantage of this, first let's tell you what its name would be. Came from an aldehyde, right? And it's an alcohol. It's called an alditol. I know. So you make an alditol. The advantage of an alditol is it makes the top and bottom the same. So in the alditol, right here, notice that the top and the bottom are the same. Now, why might I care about that? The top and the bottom are the same. What potentially could happen? No, nothing reaction-wise. Remember meso compounds? What it does is it enables the possibility, if the OHs are correctly lined up, for this thing to be a meso compound. As an aldehyde, this will never, ever be a meso compound, right? Because there's no plane of symmetry that's ever going to make that into an aldehyde, right? Impossible there. I reduce it, potentially, it could be meso. If it's meso, how would I know? How would I be able to tell? I would look at polarimetry, right? What do meso compounds do? They do nothing. They have no rotation, yeah. So what you do is you check for optical activity. So it may be meso. It may not be. That lets you narrow it down somewhat. We can kick some off the list depending on what we find out. Okay, so let's go ahead and do an example. Let's practice this for a second. So we have a little setup here. An unknown hexose gives an optically inactive alditol on treatment with sodium borohydride. Which of those sugars on your list could it be? So if it is optically inactive, what that tells us is that when it got reduced, it made a meso compound. So there has to be a plane of symmetry. So we'll look through our list and see which ones could possibly give us an internal plane of symmetry. Could it be allose? It could be, because once, remember the aldehyde's not there, it's an alcohol, okay, in this example. So allose would have a plane of symmetry right there. So it could be allose, that's a possibility. Altrose? No, not altrose. Glucose? No way. Mannose? Careful. If I go like that, is the bottom an exact reflection of the top? Well, let's go to mannose. Let's do the right one. Is the bottom an exact reflection of the top? No, because I would have to have, this OH would have to be reflected right there. So, but it's over. But yeah, it's not, no, like this? Like that? I can't. It won't work. It's tempting, but it won't work. So mannose is not okay. How about gulose? No, that won't work. Idose? No, no idose. Galactose? Could be. Yeah, right there. So galactose is another possibility. Talose? No. So look what that did for me. I know this sugar is now either allose or galactose, right? I was able to throw out the others. Now, had it been optically active, not so useful, but at least I'd be able to say, well, if it's optically active, it is not allose or galactose, right? So it does allow you to eliminate something, okay? So far now, we could tell it's allose or galactose. Beyond that, we really couldn't tell you, okay? So we'll go to our next set. Reaction with phenylhydrazine. Yes?
For the reducing reagent, you said 100 grams of metal or
LAH would do the same thing, sure. Okay, last reaction of the day. Then I'll cut you loose. Reaction with phenylhydrazine. What phenylhydrazine does is it eliminates the aldehyde. So we lose the aldehyde. Not like lose it like it flies off, it's just gone, okay? And the first OH. So we are going to turn those, the aldehyde and the first OH, turn into imines. So you can see, let's just kind of box them right here. Here they are here. And when you treat them with phenylhydrazine, here they are here. They turn into imines. So the reaction occurs at C1 and C2. We lose chirality at C2. The product has a name called an osazone. O-S-A-Z-O-N-E. It's called an osazone. And osazones, what you do is you compare the osazone of your sugar to the known osazones of other sugars. And what that does, you're basically matching the bottom 3 hydrocarbons. And so that helps you to eliminate some things too. If I know that my osazone matches these other 2 osazones, I know it's got to be one of those too. So let's take an example. An unknown sugar gives the same osazone as allose. What could it be? Well, same osazone as allose means the unknown sugar has a bottom that looks like that, right? If I have the same osazone as allose, the bottom has to look like that, because that's the same osazone allose would give. So my sugar could be allose. Allose would give the same osazone as allose, so it could be that. Look at your list. What else has the bottom 3 OHs all to the right? You're quicker than me. Bottom 3 OHs. I can't even find my list. Be faster than me. My list, there it is. Yep, allose or altrose. It's one of those 2. It's got to be. So it's allose or it's altrose. So that's another way of narrowing things down. I know it's not the others, right? Sugars that differ in their conformation at only 1 carbon are called epimers. Allose and altrose differ only at the top. It's only at 1 carbon. So these guys are epimers. And they only differ at the top carbon. Epimers is very general. You could differ somewhere in the middle. Just 1 carbon, it's called an epimer. Okay, so next time we will finish up sugars. We just have a teeny bit to go. And then we'll start amino acids, so bring that in. We'll just record it for fun. Now a glycosidic bond, if you remember, is an acetal. And we talked about that last time. So what you're going to see in a disaccharide is you are going to see the anomeric carbon involved. So it'll be the anomeric carbon of 1 sugar. And then to make an acetal, or the glycoside, we have another alcohol that has to come in, right? So just the sugar by itself, if I were to come over here and cover up this second one, okay, this is not here, and that's an OH. That's an hemiacetal for this top sugar. Cover this up. This sugar supplies an alcohol unit. Now it's an acetal. So my acetal bond, my glycosidic bond, is right here. Right there is my acetal. And the alcohol to form the acetal came from the second sugar molecule. So what we're always going to see is 1 sugar and its anomeric carbon bonded somewhere to another sugar. It could be bonded at the anomeric carbon, so you have 2 anomeric carbons stuck together. Or in this case, it wasn't. It picked this OH right here out. It could have been this OH, it could be this one, it could be that one, any of them would be fair game. So the second sugar is just bonded somewhere. Now, as far as which carbons are bonded together in this particular one, if you're counting, and let's draw the straight chain form, a real sketchy straight chain form. We have 1, 2, 3, 4 chiral carbons, CH2OH, and I don't know, there's OHs all over the place. I don't know if this is even the right sugar. I just randomly slapped OHs there. If we're counting, it's 1, 2, 3, 4, 5, 6. So, that anomeric carbon is carbon number 1. Does everyone see that? Because that's when you form the cyclic sugar, you're doing that kind of thing. So that anomeric carbon is carbon number 1. It's what was the aldehyde. So as far as the carbons that are bonded together, it is carbon 1 of sugar 1, of the first one. That's the anomeric carbon. And the second sugar, we have to now kind of count this. We have, in this case, let's go to the little highlighter. The anomeric carbon of the second sugar, right there where I'm highlighting, is number 1. Going down the chain, 2, 3, 4. Carbon number 4 is where I'm bonded. So 1, 2, 3, 4. So it is bonded to C4 of sugar 2. So what you first have to do is identify which carbons on each sugar are bonded together. Then, the orientation of carbon at carbon number 1. What we're describing there is alpha or beta. So if you're looking at the involved anomeric carbon, this one right here, is that an alpha or a beta? Yeah, the OH that was involved was down originally, and so that's an alpha linkage. So you've got to identify that too. So it is alpha. If we're going to describe the linkage, what they call it is they call it a 1 for the sugar, carbon 1, a 4 prime. The prime means the second sugar. Okay, so 1 comma 4 prime. Then it's an alpha because of the linkage being down, and it's a glycoside. I'm not going to have you guys name the disaccharide. They have really long names. I mean, it is like seriously typewritten like this long. It's crazy long. But within it, they will describe the linkage being like this. It tells you which carbons are involved on each sugar. If the link is down or up, glycoside just is a glycosidic bond. Okay, but I will not have you name them. I'd have to look that up myself. You look at the name and you're like, oh.
That name has no description of what's actually being connected.
That is correct. That is a generic, sort of a configurational description. But based on this, you could not draw that sugar. I would have to tell you that it's a glucose and a whatever-ose. I'd have to tell you the two sugars in there. If I told you, though, that you have a glucose and a, I don't know, gulose, and this is the linkage, then you could draw it, right? As long as I told you glucose was the first sugar. But yeah, based on this, it's a very generic, just like a how the things are hooked together description. That's all it is.
So does that indicate that's their interest really when they're doing war time?
Oh, I don't know. That would just describe to you how they're bonded, which could have a biological effect. There are some enzymes that can only interact with alpha linkages versus beta. So that the description of the linkage is important, but even more important would be the two sugars involved. You could stick any two sugars like that. So it will not tell you anything about the molecule in that sense. Okay, but the nomenclature is too complicated. We don't want to go there. Yes?
Did you say that acetone can be formed between two anomeric carbons?
It can be. Yeah, we're going to see an example in just a minute where that occurred. I think we are. Yeah, we are. Now, one of the questions that books love to ask you, and I may ask you on the test, is this a reducing sugar? Do you remember what a reducing sugar has? It has a hemiacetal. It needs to be able to open up at the anomeric carbon to make an aldehyde. Remember that? So it has to have a hemiacetal to be a reducing sugar. Does this one have a free hemiacetal? It doesn't have it there, right? That's not it. That's an acetal. Right there. There's a free hemiacetal. So we have our hemiacetal right here. And so it is a reducing sugar. Let's take a look at another one. Okay. This is gentiobiose. It's 2 glucose molecules together. And I'd like you guys to describe the linkage in it in terms of the same description we did before, like the 1,4' alpha, whatever, glycoside. Try to describe this linkage like that. Okay, that first glucose, what carbon atom is involved? 1. Okay, so we have 1 comma. On the second one, which one?
Prime.
6.
6.
Yeah.
Oh,
It,
6.
yeah, you see it? It's tagged up here. It's off that top CH2OH.
Yeah.
That is number 6. Yeah, that is this one down here. It's bonded to that OH. So it's a 1, 6 prime. Is it an alpha or a beta with respect to the first one? Beta, and then just glycoside.
Would the glycoside ever change?
No, I don't think so. Yeah, not for this test. In my world, no. Is it a reducing sugar?
Yes.
Yes, it is. We have ourselves a nice little hemiacetal right there. So yes, it is. Okay. You know what's interesting? A student came back who's applying to med school. He was our student 2 or 3 years ago. And he took biochem over at Davis. And it was a quarter-long class. And he said that he said that what you guys are learning right now over like this 3 days, they spent a quarter learning. They just did it like they spent a week and a half on lipids. And he said it was complete review, which boggles my mind that that could be the case. But I'm sort of, I guess they just talk real slow, yeah. Or our students are so smart that they only need a week and a half to learn the material. Yeah. The prime just means second sugar. It's the second sugar. Yeah, different sugar. It's like R prime, 2 different R groups. Yeah.
How did you tell alpha beta?
I'm looking at the anomeric carbon. The question was, how do I know alpha beta? Look at the anomeric carbon that's joined up. On the first one, it's pointed up. So it's with respect to that connecting carbon. This oxygen part, the OH, what was the OH is pointed up. So that's beta. Yeah.
Is there a way to tell which one's a prime, or you just kind of decide between
The prime, you know, the way they're always drawn like this. And the prime is always the next one to the right. And the prime is the one whose anomeric carbon is not involved. I guess maybe is the way to think about it. All right. Okay, here is sucrose. And it is formed from glucose and fructose. So it's just common table sugar is what that is right there. So this one's a little interesting because let's find the anomeric carbons first. On the glucose, the 6-membered ring one, the anomeric carbon is right there. That's an anomeric carbon. So we have anomeric carbon. Now, fructose we talked about that a little bit yesterday, or Monday, I guess. And it was a ketose. So we have a CH2OH, double bond O, and then I do not know for the life of me where the OHs are. I have to flip back and look. Oh, where is it? Where is it? I think that's it right there. OH, 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 CH2OH. So there it is. So it's a ketose. Now if you look at this thing, it closed up into a 5-membered ring. So it's a little bit different looking. And what went on with it was, if this guy swings around, closes in there, that's the oxygen's counting number 1. 1, 2, 3, 4, 5. So that happened. So that means that the molecule, when you draw it in cyclic form, is going to have 2 CH2OHs hanging off of it. One from the top, one from the bottom. Everyone okay with that? All right. So our job, and I think we did this last time, I have a vague memory of this, is finding its anomeric carbon, which is not quite so obvious in this case. So the anomeric carbon is going to be the one that was the ketone. So it'll be that carbon right there. It will have an OH on it from the electrons coming up on the ketone. That will form an OH. And it will have a CH2OH. So looking at this guy, I see 2 places where there's a CH2OH. One is at the far right-hand side, right there. But there's no OH off of it. So that one is not my anomeric carbon. That means that one had to be. Is everyone okay with that? All right, so let's go ahead and write on this. That right there is the anomeric carbon. So this kind of answers Aspram's question, can you ever have 2 anomeric carbons bonded together? Yes, you can. So now describing the linkage in this thing. It's a 1, 2, 1 to 1 or 1 to 2? 1 to 1 is the first one. I'll agree with you there. What carbon number is bonded in that 5-membered ring, the furanose ring? It's bonded through the anomeric carbon, right?
Yeah.
Which is carbon number. Come on. Okay. There's 1. There's 2. Right? 2. Pretend you were naming it like IUPAC. This would be 1. It would be a 2 whatever own, right? So it is carbon number 2. So it is 1 and then 2 prime. And now what? Alpha glycoside. Is it a reducing sugar? How come? There is no free anomeric carbon. There is no hemiacetal. Okay, no hemiacetal. Yeah?
How did you figure out it's alpha?
What I did to do alpha is I went off of the first sugar right here. This is an alpha linkage.
Is it always the first sugar or is it always the second?
You know, the ones that I have seen, it's always been the first sugar. I asked one of our teachers who does biochem, I was asking her something like, well, what if you had, 2 anomeric carbons together, one's alpha, one's beta, what do you do? So I didn't know the answer. And she said a lot of times what they'll do is they'll just leave this off since you really can't put it there. Okay, so don't sit there, don't panic. But I go off the first sugar.
Alpha, beta.
Yeah, it could be, yeah, it could be. It just depends how that second sugar is oriented. So I won't give you guys anything that weird.
Second sugar is beta, and the first one is alpha. And biochem,
The ones that, what I have seen is they give it in reference to, I don't know why that is, the first sugar. But like I was saying, what she said is if it's ambiguous, then you will see sometimes that is just not included at all. It's a 1,2 prime glycoside.
or both.
I haven't seen it with both, but that don't.
They teach us.
Yeah, do they teach you just to put both? Okay. Yeah, that would work. So you would put 1, then 1 alpha, 2 beta, like this?
No, 1 and 2 prime, alpha, beta, glycine.
Okay. 1 and 2 prime alpha, beta, glycine. That makes sense. You specify both. All right. Let's move on to bigger things. Polysaccharides. This is where you have essentially a sugar polymer. So it is a whole bunch of monosaccharides, little cyclic units that are linked up together. So we're talking there like tens to thousands of little units like that. This particular example that I have here is amylose, where it says N is 1,000 to 6,000. That little n is referring to that right there. So you know how when we have a long molecule, we go CH3, CH2 in parentheses, and we might put CH2 because we don't want to go CH2, CH2, CH2. We just put a parentheses and a 10. That's telling you how many units are there. So it could have 6,000 little sugar glucose units stuck in there in this chain. It's hugely long. The glucose molecules are all linked the same. They're all linked by 1,4' alpha glycosides. So the linkages are the same. This thing just repeats. This is a component of starch. So amylose is found in starch, potatoes, grain, corn. And we have enzymes that can break down the alpha linkages and can then turn this thing into sugar molecules. It turns it into glucose. We get little glucose units in our bodies. One of the interesting experiments they'll do, I think they do it in biology, and in Chem 309, I think they do it. It's kind of disgusting, as I recall the experiment. They take a saltine cracker, chew it up, let it sit in your mouth and kind of ferment in your spit. And that's the disgusting part. And what will happen, saltine cracker is not sweet in and of itself. At first, it's just kind of like a nothing. But after it sits for a while, it starts to taste sweet. What's going on there? The enzymes in your spit are breaking that down into sugar. And then what they do is they spit it out into like a watch glass, and they test it with, I guess, iodine, and it turns purple in the presence of starch. And so the cracker, when you can smush it up and it's not spit upon, it's not purple. But after it sits in your mouth for 20 minutes and gets worked up, then you spit it out, it turns purple. And it breaks down the starch into little glucoses. They test it somehow. But it'll taste sweet. Try it at home. I've done it. I didn't believe them. And I chewed a cracker up and let it sit for 20 minutes, and it did kind of taste sweet. And it was not good. Another thing, I was reading about back when we were in the war in Afghanistan was like just starting, and mountain villages had gotten cut off because of the war from food supplies. And they, up in the mountains, it's very barren and there's like no soil. So they really have no way of growing food in particular. And so they were starving because they didn't have food. And what they were doing is they were taking and grinding up grass and making for their kids grass pancakes or something like that, because it would fill the kids up, because just it's a filler, but the kids were starving and dying. And that's because, as we'll see in just a minute, grass, which is cellulose, doesn't have alpha glycoside linkages. And our bodies can break down alpha glycosides, and that's it. And so they're eating this cellulose, this grass, and it was never breaking that unit down into sugar, which meant they were never getting any energy and starving.
They make wheatgrass juice.
They do, yeah. They make the.
Sell it like crazy.
They do, but I think the cellulose is like the rigid plant structure, so they're squeezing other stuff out of there, I think, because if you ever look at them make it, they always throw something in the trash that's the reedy, grassy part. Yeah.
They make a bacterial pill that you can take that will break apart.
Okay, so so the bacteria can break the beta
So if
linkages.
you eat grass,
Okay.
you can if you have it.
You can if you had to. Yeah, that makes sense. I understand too. There's a bacteria in cows' stomachs that help it to break down those beta linkages.
They also have two stomachs.
They do. Apparently, it's not really efficient, so it has to get worked over quite a bit. So, okay, that brings us to cellulose. Cellulose is again really, really long. It looks the same, and it kind of is. It's a bunch of glucoses. They're linked 1,4', but here's the difference. Cellulose is a beta linkage, and it's much more rigid. It gives structural rigidity to plants. but we generally cannot digest it. And like I said, the animals that graze have bacteria that can do it in their stomachs, but they have, like cows have what, three stomachs? Two, and so it takes a while. Also, I was reading somewhere a fisher bug. I don't know what a fisher bug is, but a fisher bug can break apart beta linkages. Bit of trivia. All right, that is it for carbohydrates. All right, any questions? All right, let's do amino acids then.